Howdy, my name is Adamari Cerda and my partner Mario Flores Castro and I will be discussing our 10th experimental lab report on the PCR experiment. To begin, PCR is an experimental method that allows for the amplification of genetic material and increases a given quantity for genomic analysis. This experiment focuses on using PCR to detect and amplify E. coli DNA. This method ultimately supplements food safety measures to prevent foodborne illnesses. To begin, the materials needed are a PCR thermocycler, PCR tubes, the E. coli bacterium, PCR master mix, forward and reverse primers, nucleus free water, positive control DNA, negative control, electrophoresis and gel equipment, DNA ladder, gel loading dye, and agarose gel. In order to begin with this investigation, we will begin with the master mix prep, and we will be obtaining 67.5 microliters of the master mix and 45 microliters of the primer mix. We will then aliquot evenly into each of the four PCR tubes. In this case, it is 25 microliters of the master mix into each tube. Then we will be adding in 5 microliters of DNA sample into each tube before vortexing. The PCR amplification portion of the investigation calls for placing the tubes into a thermocycler. Then input the following steps. The initial denaturation step calls for being placed at 94 degrees Celsius for 3 minutes. Denaturation at 94 degrees Celsius at 30 seconds. The annealing step is at 57 degrees Celsius at 30 seconds. The extension is placed at 72 degrees Celsius at 30 seconds. The number of cycles will be 35, and the final extension is at 72 degrees Celsius at 60 seconds. The gel prep portion of the investigation calls for adding 0.75 grams of agarose into a clean flask. Then measure and pour out 50 milliliters of 1xt AE buffer. Heat in the microwave for 1 minute and allow the solution to cool, and then add 5 microliters of cybergreen. Once loaded onto a gel dye, the samples are then visualized by gel under a UV light, where the results will ultimately be recorded for further analysis.
These were the results of our gel electrophoresis. From order of left to right, we have first the ladder, then sample A, then sample B, then our positive control and our negative control. We saw how in sample A and B there's the presence of bacteria, the bacterial DNA, and it was amplified. It's important to know that our negative control also showed a positive band, meaning that it might have been contaminated. From this, we arrive to the conclusion that the presence of bacterial genetic materials present on the food samples and the negative control was possibly contaminated. This is important because it is useful to test food safety and to check for the presence of bacteria. Thank you.